Hello. Well, my name is Gemma, PhD student at the Institute of Evolutionary Biology in Barcelona, and I will talk about how we can illuminate the functional landscape of the functional dark proteome across the animal tree of life through natural language processing models. It will be more of applications. Working. Yeah.
Oh, it was turned off.
Oh, okay.
Wait. And I click, okay. Just a second. Δεν το βλέπω. Γιατί του έκανε ένα κλικ. Ναι, σε ευχαριστώ.
Thank you. So yeah. So how we can illuminate the functional landscape of the across the animal tree of life. Okay. So adaptation and function are like two aspects of one problem. So let's imagine we have this phenotype. In this case, it's related to the wing color, and this phenotype can be adaptive or not. If we look at the genetic basis of this, we may find that it's caused by this gene. But in order to fully understand how this gene is related to the phenotype and the adaptation, it's important. It helps to know the function of this gene. And only with this, we can have a fully picture of how the evolutionary process works from the molecular to the phenotype level. So in order to know the function of genes, they created this gene ontology, which is a set of control vocabulary that assigns functions in terms of words. It can be divided into categories such as cellular components. So it tells that for this protein, for example, it's located in the mitochondrial membrane. Molecular function, what's the function itself of the molecule or the biological process it's involved with. So traditionally, the ideal thing to do would be to use experiments to identify these functions, and then for each protein, you would say, okay, this is involved in this, and this process. But especially in evolutionary biology, you cannot do that. You cannot do a lot of experiments. So we have to rely on bioinformatic prediction of these functions. And what we usually do is use tools such as BLAST to annotate them based on the sequence similarity, based on the homology that they have. So from our protein sequences, we compare them through a difference, and we transfer the gene ontology terms, so that if this protein is similar to another, it has the geo terms, so we assume that it has the same ones. One example of these tools is EGNOG Mapper, which is widely used for non-model organisms especially. The problem is that based on homology or sequence similarity, it has several limitations. So for example, this is a tardigrade species, one animal. There are like 40% of the genes that you cannot find any homologous similarity to the reference databases. But this problem also extends to model organisms, that there are still a percentage of genes in model organisms that we don't know what their function is. So in not only this, a percentage could be larger or smaller of the genes in the genomes that are dark, that we don't know what their function is. And with the rise of all these genomic initiatives to sequence the genome of a lot of species, this problem is getting worse and worse and worse because the scale of the experiments cannot cope with this speed. So, why are these genes not annotated? It could be based on homology, it could be because these lineages are not in the databases. So, for example, here, the marine biodiversity, most of them are arthropods, but most of the genome sequence are from fish, for example. So it could be that you do not find a match because you don't have a reference. Could be of the limitation of the method itself, so that it could be that they are homologous, so they have supposedly the same function, but you cannot identify them because they are too small or because they are highly evolved, highly diverse. so you cannot identify them. Or that could be de novo proteins, so they just appear de novo, so you don't have anything to compare them with. So I will talk about exploring alternative methods for this. The first one will be based on machine learning or deep learning, and I will briefly focus, explain a bit this. So there are several methods, but I will work with one that is called DeepGo Plus, which is trained using a convolutional neural network and based on sequence similarity. So for people like me that are not very don't know a lot of machine learning, for people that know less than me for molecular biology, you just give the list of proteins and you get the gene ontology term. So it's pretty convenient. But there are other alternatives based on natural language processes. In this, I will briefly explain this with this example of words. So you have several words, and in these methods, you that they have been trained with other data sets, you get the vector representation of them called embeddings. And in the embedding space, you can see that the words that are more similar in their meaning, they are closer to each other. So we can transfer this with proteins and what we call protein language models. So you get these sequences, your sequence of interest and the references, you get the embeddings using these protein language models. And in the embedding space, you can, from your protein of interest, transfer the geo terms from the closest in the embedding space from your reference, that you know the function of those. I will talk about one that is called GoPretSim, and I've applied two different methods, SecVec, which is based on Elmo, and Prot5, which is similar to the T5 models out there. Okay, so the idea is we have the based on homology, based on deep learning, and this one based on natural language processing, and all these two methods of deep learning and natural protein language models, they have been benchmarked using just a very small data set. So we are wondering how do they work if we give to them a whole list of proteins of one organism. So we did a benchmarking using model organisms. I will not talk much about this. But basically, with model organisms, as I told you before, there is still a percentage that is not annotated. So we took the proteomes from UniProt and their annotations, and we compare how are the predictions we get using the protein language models and the deep learning. And you can see here, basically, a bit of a spoiler of what I will show you for non-model organisms. It's that with deep learning, you get more predictions, but compared to not protein language models, but the precision, how informative the terms are, how similar they are to the gold standard, and if we can recover functions from transcriptomic studies, for example, it's much better when we apply protein language models. So, as in my lab, we are working with weird organisms, non-model organisms. We wonder, how do these methods work with non-model organisms? So I have this huge data set of animals. It has 970 species, 960 animals from genomics and transcriptomics. It's 23.5 million genes. They are all here across it includes the whole diversity of animals, not only the ones that are most known, but some weird ones. Okay, so this is a small, like a snapshot of this. This is the annotations using the method based on homology of the proportion of genes that are annotated by that. So you can see that depending on these are the animal phyla. So depending on the phylum that you are looking at, the proportion is different. With vertebrates, somewhere here, yes, are the ones that have a higher proportion of genes annotated because they are more studied. But there is a huge diversity on that, and in some species, you lose like more than half of the genes without annotation. So if you want to do a prediction there of an evolutionary study based on the functions, even a statistical method or so, you are losing half of the information that you could get if you just use homology. So first of all, regarding the proportion of genes that are annotated, this is the based on homology. We get half of our 23.5 million genes annotated, but with the other three, we get all of them, almost all of them. If we make some filterings, one based on the depth inside this ontology of inside the gene ontology, and another one based on reliability index, which is like a threshold they put on the original papers, based on how you can how good is the prediction, how you can rely on that. So we can see that It reduces a bit, especially for GoPretSim segment, but still, especially the Go Plus and GoPretSim with Prot5 model, the proportion of genes that are annotated is much higher than based on homology. Okay. Another interesting thing is that the number of geoterms that you get is much higher when you use deep learning and, well, a bit homology compared to the natural language processing. And this, depending on the objective that you have, but if you have like a lot of data, for example, having a huge file with a lot of gene ontology terms that most of them are not informative for you, it creates more noise for the analysis. And it's also like increases size of your files, and for parsing it for later analysis, it's much harder to do that. And if you need the other terms, you can even go back to the gene ontology. And the final thing I will talk about more of how good are these is based on the information content, which is a measure of how informative are these terms based on the gene ontology. So imagine we have this term, DNA damage response in here. So this one is more informative than the response to stimulus because it's more specific. So for some biological process, this doesn't give us any information. So it's better to have genes that are more specific because you can know more what this gene is involved in. So I will just show you this for biological process, but if you look at the density plot of the information content in Vigo Plus, most of the terms have information content that is quite low, meaning that they are not very specific and are not really informative compared to the other three that have they are much more informative. Well, the same for molecular function and cellular component. I will skip this part. Okay. There are more analysis in the paper, but I think I will skip them a bit, but you can check it later if you have any questions or so. Well, as I'm using tools that were available that were developed by other people, in the case of GoPretSim, using the Prot5 model, which I've shown you that this and in the benchmark that we did with model organisms, they are supposedly, well, better than the machine learning ones. There is this web server to apply it, but the problem is, like me, I have 24 million genes almost. I cannot use the web server. So if we have created this pipeline called Fantasia, so that for people that want to annotate a whole proteome, you just give the list of proteins. It applies this GoPretSim using this model, and you get the gene ontology annotation of all your genes, and are ready to use it for you for gene ontology enrichment or other stuff. As well as it's one good thing is that it can be run with GPUs and the execution time decreases a lot. Mainly, the main problem it has is that it relies it's the usage of memory because there's a linear depending to the number of sequences. So the more sequences you have, the more memory you would need to. But for example, people that work with genome assembly, they already need a lot of memory for doing the assembly of the genome. So that for them, it would not be a problem, for example. Okay, so now as I'm more a biologist, I want to talk to you a bit about how we have applied this to discover in the animals what are the functions that were not annotated before using homology. So what were we losing in that, and if it's informative or not. So we have for all our almost 1,000 animal species the functions of this, and we perform some gene ontology enrichment. So we have this is our genes that have been annotated, and only these ones were annotated based on homology. So we perform a gene ontology enrichment of the genes that were not annotated by homology compared to the background. We did it per species using a software called TopGo, which takes into account the topology of the gene ontology. Okay. So we took the geoterms that were common between different species and the ones that were not common, and we performed some meta-analysis to obtain a gene ontology treatment per phylum, because you cannot do that with the size of my dataset altogether. And with that, we obtained the significant gene ontology terms per phylum, and we repeated that 29 times for each of the animal phyla. And then we looked at the functions. So first of all, we found some functions of gene ontology terms that were common to all animals, and they were related to the response, viral response, transposable elements, immunity, or toxin activity, which are quite relevant for the biology of animals. Then for each of the phylum, we like cluster the geoterms together to make, instead of having the specific geoterms, to make a more general category to know more or less where they fall. And this is just a subset of the results. So for example, by biological process response to a stimulus, there are a lot of geoterms that are enriched in response to a stimulus, to the environment or to other organisms. Others involved in immune system, the development, signaling, reproduction. In the case of molecular function, there are many that have catalytic activity, so they would be enzymes or transporters, and most of them in the category of cellular component, which is in which part of the cell they are, they form protein complexes. So, well, of course, this is just like a snapshot. If you wanted to focus on one specific protein, you would have to go deeper. But as a first glance of what am I missing of this organism, of I want to know what is this, it's really interesting to have this. So I will just one example of one phylum. So I don't know if you know of tardigrades. These are these microscopic small animals that can survive in very extreme environments, including the space. So these are, this is a tree map of the representation of the gene ontology functions that are enriched. And one interesting one is the detection of a stimulus. So we have terms related to response to UV light, water deprivation. So for these animals that are very resistant, we found some geoterms that are involved in these functions that make them so special. So for future studies, it would be interesting to check these putative genes involved in this one. Even just a very specific example, there is this protein that has these geoterms related to damage response in this species. There is one homologue in another species which was not annotated in UniProt, but with our methodology, we were able to recover exactly the same geoterms that this one has. In fact, this one is an intrinsically disordered protein, and we showed that in our whole data set, the ones that are mostly disordered and globular proteins, that are more flexible, are significantly less annotated by homology-based method. And besides the soup, there are also some examples in Drosophila melanogaster, such as this one here. So just like to summarize, natural language processes outperform other methods like deep learning because they have more annotated genes, they have less number of geoterms, and they are more informative. One thing I didn't talk about, that there's no difference when you use just the longest isoform for each gene, or if you use all of them together. I presented to you this pipeline Fantasia, and it can be a potential tool for revealing hidden functions, or at least a starting point to know these hidden functions. So all in all, even if the darkness of genes, we may find relevant functions, if we have a way to turn on the light. Thank you so much for your attention. Thank the collaborations. And you can check the preprint. Yes.
Thanks a lot.